The Helmholtz-Center for Infection Research in Braunschweig regularly bestows the Paper of the Month award. The prize honors an outstanding paper published by a PhD student or a postdoc. The awardees for August 2014 are Christian Meyer and Peyman Ghorbani, who work at the Institute for Infection Immunology, led by Tim Sparwasser at the Twincore in Hanover.
Hi, my name is Peyman Ghorbani. I come from the lab of Tim Sparwasser in Twincore at the Medical School Hanover, and I study infection immunology. So in the paper we studied the development of CD103 positive dendritic cells, which are the key drivers of intracellular targeted immunity. And we identified a novel culture protocol to efficiently generate large numbers of these dendritic cells by combining two growth factors, FLT3 ligand and GM-CSF, which were previously only used separately to generate in vitro dendritic cells. So these CD103 positive dendritic cells are very difficult to find in the mouse. There's very few numbers of them, and if you isolated all the dendritic cells that were in the lungs and livers and intestines, you wouldn't get really enough to use in vitro assays or to inject them back into a mouse to check for the effects of a vaccine, for example. But using this method, we can isolate the bone marrow from a mouse and generate roughly 30 million dendritic cells just from one single mouse, which was previously not possible. And using this method, we can generate large numbers of these dendritic cells to study them for the effects of different toll-like receptor ligands or other stimulus on their activation status or their capacity to uptake bacteria, for example. Mm-hmm. So the next thing to look at with these dendritic cell cultures is to see whether there are different factors that can be added or compounds that could be added that influence the differentiation of these dendritic cells towards a tolerogenic phenotype or a more immunogenic phenotype for vaccines, for example. And this is interesting in the context of immunotherapy, where dendritic cells can be pulsed or primed with a certain antigen, and then these dendritic cells can then be injected into a mouse to protect them against cancer or bacteria or virus. And why this is interesting is because this is a really hot topic in the clinical field for cancer immunotherapy in humans, and these dendritic cells also can't be cultured so efficiently from human blood, for example. And using this method, we might be able to identify certain features of the culture that are important to generate the same dendritic cells in humans. We identified a novel culture protocol to generate really large numbers of CD103 positive dendritic cells from mouse bone marrow. And these dendritic cells closely resemble what we can find in mice.